transmitter. The illustration of this is the dopamine-associated currents. I pulled this particular article because of the next slide, but I just want you to see that in this hex cell in which the dopamine transporter has been expressed, when stepping from a holding potential of minus 60 millivolts to one much more hyperpolarized, you can see the holding current has an additional component to it in the presence of dopamine. So the difference between these two steady state levels would be the current associated with ions moving through the dopamine transporter. These currents can be mimicked by amphetamine because amphetamines bind to the dopamine transporter and activate it, and that can be then blocked or prevented by cocaine, which also binds to the dopamine transporter, but instead it acts as an antagonist to it. So two very important street drugs have direct action on the central nervous system and synapse by binding to dopamine transporters. And that is their principal mode of action in terms of the high producing effects of these drugs. This dopamine current is not chloride, as is true in many of the other transporters, say the norepinephrine transporter or the excitatory amino acid transporters. Here it's shown that the ion responsible is sodium, because if one removes all of the sodium and replaces it with choline, this current goes away in the presence of dopamine. We have specific antagonists that act at the dopamine transporter to block the dopamine-induced current. And this is one called GBR, and then there's a series of numbers. So what might be the function of these transporter-associated currents? These experiments are now in cultures of midbrain dopaminergic neurons. So they have not only the transporter, but they have dopamine receptors in them. And you can see the spontaneous rate of firing becomes inhibited when dopamine is added to the culture. One blocks the D2 receptors with sulpiride. The action potential firing is restored, and it's actually a higher frequency than it was initially. And that high frequency is blocked if one adds cocaine to block the transporter. In B, all of the various dopamine receptors are blocked. So in this condition, adding dopamine doesn't hyperpolarize, which is the response of the D2 receptor, but rather one sees again this increase in frequency of firing that is blocked when the antagonist for the dopamine transporter, this GBR compound, is added. The bottom two graphs show the same data. What's on the y-axis is just the frequency of firing as opposed to the raw traces. So you can see dopamine stimulating the frequency of firing and the dopamine transporter antagonists blocking it, or amphetamine increasing the frequency of firing and the dopamine transporter antagonizing it. So it may be that these extra currents carried by neurotransmitter transporters may be modulating frequency of firing, spontaneous activity, or possibly even the consequences of synaptic activity. So then, in summary, what are all the possible roles for these transporter-associated currents? Many of them, since they transport chloride more than what we just saw as sodium, may actually function to bring chloride into the cytosol or into the vesicle. Often chloride is used to balance some increased acidity from some other cellular process. We've seen the data that suggests they could modulate the frequency of firing and or synaptic transmission. They may be necessary for some kind of ionic balance, maybe not the ionic changes from the transporter operation itself, but some other coupled activity within inside the cell. Or they could provide a driving force for other linked cellular functions.
Lastly, we want to consider whether or not transporter uptake of neurotransmitter is important in clearing the concentration of neurotransmitter in the cleft and terminating synaptic transmission. In this very careful and exquisite biophysical experiment by Susan Amara and colleagues, she measured the number of charges carried by a single glutamate transporter in one cycle, and that varied with membrane potential. And she also measured how many times a single transporter molecule cycled in any 1 second. And using these two measurements, she was able to calculate that the cycle time for a single transporter molecule to move a single molecule of glutamate was 70 milliseconds, which is much slower than the 1 or 2 milliseconds needed for the synaptic decay of a synaptic potential, postsynaptic potential. And from this, they reasoned that the transporters were not important for clearing the cleft of synaptic neurotransmitter, but rather the binding of the neurotransmitter